I'm very excited to be here today and share with you more about what it is we do at Kvantify, or Kvantify we say in English. So Kvantify is a relatively new company. We've been around for a little bit more than 2 years. We're based out of Denmark, but now also have an office in London. We've grown extremely rapidly, so we're now a team of 65 people, and everything we do is software and algorithms trying to do useful things with quantum computing. Our mission, our vision, is to solve difficult problems. And we don't solve difficult problems just for the sake of solving them. We do it to create value. So it's a laser-sharp focus on what will actually be valuable, and then using the available tool set that we have today and will have in the future to sort of achieve that mission. And we work with quantum computing, but not alone. So our view is that if you're going to do anything useful with a quantum computer, you're going to need a classical computer as well. And our picture is this, we call it the onion model. We didn't invent it, but it sort of illustrates how we think. So if you have a quantum algorithm, it sits in the core and it does what quantum algorithms do, but it's not enough. You need to wrap it in classical methods that increase in accuracy as you get closer to where the quantum magic happens, if you will. And this means there's 2 parts you need to work on. You need to work on the quantum algorithm. How do we squeeze the most we can of power out of existing and near-term hardware and get as much as we can there? And then where do we apply it? What industrial problem of relevance do we apply the quantum power to, and then build the other rings, if you will, the classical approaches that sits outside of quantum? And the logic is that quantum is extremely accurate, and with the current theory, you can simulate maybe 20 electron orbitals on 40 qubits. So what can you use 20 electron orbitals for? And then you build classical methods that are close in accuracy, so quantum chemistry, if you will, on classical hardware, and then it moves out and becomes less and less accurate as we move away. That allows us to gain speed and still do useful things. So we work on both, and we try to match them up, and one of the things we do is look at molecules. So if you can simulate 20 orbitals on a quantum computer, one of the places we are quite confident we can add value is in quantum chemistry, and one of the very interesting places for quantum chemistry is drug discovery. So the picture you see is of a protein and a ligand, a small molecule interacting, and it's a little bit, the drawing is slightly off, but you can sort of see you use a quantum computer right where the magic is happening. And far away you can use mean field methods, DFT or whatever, and stitching that all together is exactly what we do to try and do useful things. Now if we talk about quantum, so we zoom in on the center here. Most of you have heard about or know VQE. It's a variation algorithm that you can run on NISQ hardware. Again, you can simulate 20 electron orbitals on 40 qubits, but you need a lot of gates, which makes it practically not applicable for anything. The previous industry standard to do useful things was an algorithm called adapt VQE, and you pay for the usefulness of adapt. You get fewer gates, but you have to add a lot of measurements. That means that if you have n qubits, you need to have n to the 4th plus n to the 8th measurements to do your calculation with adapt. That's a lot, and that makes it hard to sort of have adapt converge. Our algorithm called fast VQE, which we published last year, reduces the n to the 8th. So we're down to n to the power of 4 plus 1 measurement in the circuit. And that basically does that if you run, this is on H4, so the theoretical molecule H4, it makes the calculation for electron structure on H4 converge on existing hardware. So you can see the red line is adapt, and the dotted line is what's called chemical accuracy, 10 to the minus 3 heart rate. This is where you want to go. And adapt doesn't really go to that, but fast VQE does. And that's due to the elimination of measurements. It also means that fast will be a useful algorithm for quantum chemistry with 40-ish qubits and then 100,000 CNOTs, which is still quite a lot. But this you can read more about. We've published a few papers on it. And what we're doing right now is, this is a benchmark case, right? What can we use this for? And we're working with a few companies on enzyme simulations or battery chemistry, aside from drug discovery, as you see here, to apply quantum computing on relevant chemical systems. We're still not at advantage, but we're pushing what can be done on existing hardware. Now, where do we go from here? Well, there's still this n to the power of 4 that we need to get rid of. And then could we squeeze more information into the qubits? Could we, instead of 20 orbitals in 40 qubits, have 40 orbitals in 40 qubits? We think so. And that's our next algorithm, called Beast VQE. So with Beast, we will have useful chemistry on quantum computing hardware with 40 qubits and 1,000 gates. Still not there, but we're much closer than we've ever been to having something that actually works for useful chemistry on quantum computing hardware. So what do we use it for? Well, as I mentioned, we're very excited about small molecule chemistry, and in particular drug discovery, because it's an area where small molecule chemistry really has a lot of value if we can get better at it. Existing drug development cycles take more than 10 years to go from an idea to actually having a drug on the market. It costs you more than $2.5 billion to go there. And then when you enter clinical trials, you still have a 90% failure rate. It's part of the explanation why new drugs are so expensive, and also we're not very successful at bringing a lot of new drugs to market. Drug discovery, in essence, is a funnel. You start trying to figure out, if you have a disease, which protein is this disease coupled to. That's a process called target ID. It's a very good case for machine learning. You need a lot of data to sort of identify. It's not quantum at all. So we start below that, basically. But then you go from, we have a protein, we want to inhibit it with some molecule. You try starting with a lot of molecule, and then you iterate through the funnel, ending up with a few you take to clinical trials. And hopefully you take one of them to market. Through this iteration process, you do more and more advanced methods and apply more and more accurate both computational methods and lab experiments to try and understand the effect and efficacy and safety of the molecules that you're looking at. We focus on, after target ID, anything before preclinical. It takes you 3 to 5 years today to go from target ID and until you actually go to clinical trials. We want to reduce that to less than a year, which requires us to do quite a few things on the computational side, but we do believe this is possible within a reasonable time frame. And then while we do that, we want to address the major causes of failure of drugs when they then do enter into the clinic. Typically, we most of the time, or sometimes we don't know what's going on, but when we do, it's related to dosing or efficacy or safety of the drugs. At least those are the major causes. And we can address those if we can better understand the chemistry early in the funnel. So our initial focus has been a topic that's called unbinding kinetics. So unbinding kinetics is, how does this small molecule get away, basically? So now you have a protein, you know this is related to a disease. You even found a molecule that can stick to the protein in a pocket. It sticks, but how long does it stick? Obviously, you need to have it there for a while for it to have any effect. So if you understand unbinding kinetics, you can have better selectivity in terms of you need it to stick to the right molecules for the right amount of time. You can better estimate what dosage you need. If it sticks around for a minute or an hour or a day or a month, there's obviously a difference in how much you need to put into the body for it to have an effect. And ultimately, understanding unbinding kinetics can help you reduce clinical failure rates. Now today, unbinding kinetics has very limited availability. There's no computational solution commercially available. You do it in labs. And when you do it on a computer, it takes you a long time. Hundreds of hours of GPU time to do unbinding kinetics computationally. Well, until now.
Let me introduce the first outcome of our product development, a groundbreaking drug discovery tool. This is Kvantify's initial step towards revolutionizing computational drug discovery.
Life sciences and drug discovery in particular is one of our key focus areas, because that's where we expect quantum computing to bring enormous value. Drug discovery can be a slow process, not to mention expensive, with failure rates above 90% and drugs usually taking over 10 years to reach the market. We believe there's a large potential to be uncovered. When it comes to finding and evaluating drug candidates in silico, precision is essential. And with quantum computers, we get new possibilities that go beyond what we've previously been able to achieve. In silico, calculations have already made a difference, but new quantum-based methods will be the key to really disrupt the drug discovery industry. Our vision is to develop quantum-ready state-of-the-art software for drug discovery. At Kvantify, we're convinced that combining classical and quantum computing methods is the future. Classical methods will deliver speed, quantum computers will deliver precision. That doesn't mean that the classical methods should remain as they are. They need to be tailored to integrate quantum algorithms, and this is exactly what we've done for our unbinding kinetics tool.
We have developed a novel method to predict unbinding kinetics, or K-off rates, a key metric indicating how long a drug interacts with the target, something that has direct impact on critical considerations such as dosing. Current computational methods rely on molecular dynamics. The approximations stemming from force field-based approaches like MD limits the accuracy, and MD is also a slow method requiring complex setup. Our method is an entirely new approach that has no comparable commercial alternative on the market, and today many pharma and biotech companies measure unbinding kinetics only for very advanced molecules or do not measure it at all due to the complexity, the cost, and the timelines not being compatible with drug design cycles. As a result, this remains an area of drug discovery that still heavily relies on trial and error. We have performed thorough benchmarking on literature data to demonstrate predictability across multiple protein targets and diverse ligand series. Using our in-house cheminformatics expertise, we have benchmarked across 7 protein targets with data for approximately 130 ligands. The proteins include popular drug targets such as kinases, GPCRs, as well as molecular chaperones. When we compare our calculated unbinding kinetics values with experimental data, we achieve excellent correlation. The method is also rapid and highly scalable. This allows the selection and optimization of small molecules based on kinetic properties to be data-driven rather than being based on trial and error, and to be done earlier, de-risking the entire drug discovery process.
Our new kinetic solution enables faster and more accurate screening than is currently achievable in silico. What we're launching today is the first commercial solution for simulating unbinding kinetics. With this solution, we will enable pharma companies and biotechs to reduce the risks involved in the drug discovery process. Time is money, and our new solution will save lots of it. For example, it makes calculations for one compound more than 100 times faster than current alternatives. That means that it'll be ready in the time it takes to make one single cup of coffee. So what better name for our very first Kvantify product than to call it Koffee? With the introduction of Koffee, we have re-engineered the problem of unbinding kinetics to create a groundbreaking state-of-the-art solution. We've done extensive benchmarking to show that Koffee delivers accuracy that is similar to experiments. It also has an algorithm that can be used for a diverse set of ligand and protein targets. When it comes to performance, Koffee goes beyond what's currently possible. And we've also made sure that it's quantum ready for the future. We're currently looking for strategic partners that will get early access to Koffee. So if you want to be part of the drug discovery revolution, get in touch.
There you have it. Kvantify Koffee. Our first product that does something that has never been done before, never been possible on classical compute, and ready for the future on quantum. Unbinding kinetics in the time it takes to brew a cup of coffee. Now, just to reiterate a few of the points from the video, Koffee works across a very diverse range of targets and ligands. We have benchmarked also across or outside of these targets on the slide, and we have yet to find a combination where it doesn't work. It can detect very subtle molecular changes. So if you look at these two molecules, there's only one aromatic ring difference between them. But they obviously have quite different K-off rates, which you can see in experiments, but it's also verified when we compute them using Koffee. And then Koffee is quantum ready. It's engineered in a way that it can leverage the power of quantum computing from day one of useful quantum computing. It also means that we're moving away. This is a method that is typically done by what's called molecular dynamics, and we're doing something different, which we're quite excited about. So, in essence, Koffee offers a 100 times speed up from what is the industry standard. It's validated by experiments, and it is applicable across a range of targets and ligands, and it's quantum ready. So the power of Koffee is delivered today, and today it delivers unmatched speed. When we boot up the quantum computer, it will also deliver unmatched accuracy. So that allows you to iterate smarter as a drug discovery company. You can save time and money. You can apply or investigate unbinding kinetics much earlier in the drug discovery funnel. And instead of doing it in labs when you go to preclinical trials, you can do it with high throughput screening very early and become much smarter at selecting which compounds you take forward, de-risking the entire drug discovery process. So what's next? Well, Kvantify Koffee will be broadly available September 1st, but if you want to get ahead of the queue, we're looking for a couple of strategic partners that will help us push it all the way to commercial launch on September 1st. And then we're not just doing unbinding kinetics. We're also doing docking and binding affinity, the two other items that are really at the core of computational chemistry and the drug discovery workflow. And both of those will be launching this year. And both of those will see similar improvements to what we've just seen on unbinding kinetics. But that will be for another place and another time. So with this, thank you for your time today.
Okay, we have time for a question or two. So first question, can your unbinding tool, Koffee, I guess, can it be used for other chemical unbinding problems other than drug discovery? Are there other material applications that it might apply to?
Yes. The short answer is yes. We are looking at other applications. You can use it for enzyme binding on surfaces. There's a few other places as well we're investigating, and then the method in itself could also, and that's another thing, prove valuable outside of unbinding in general. So,
yes. Okay. Next question was about what types of quantum computers are you running on or planning on running on in the future? As a software company, do you have any preference at this time, etc.?
Our clear preference is for a quantum computer that works, which means if we want to go to the useful quantum computing stuff, we need two gate fidelities around 4 nines, which is still a little bit away from current hardware, but 40 qubits, 2 qubit fidelity around 4 nines, and then 1,000 gates and we're in business. We run, Steve, who sits in the audience, said to me a few hours ago, I want to get my hands on everything I can get my hands on. So we do run on all the hardware we can get access to. We work extensively with Microsoft and Amazon and their cloud offerings, as well as 10, I guess, hardware providers individually to test on all the different platforms and see what works and what we can do. But we, as a software company, we really just need one that works.
Okay. Okay, and any suggestions on what types of companies, you're opening up kind of initial access to selected customers. Can you specify what types of companies that might be?
It needs to be early drug discovery companies. So at the stage we are at now, we're trying to verify that we can reproduce a broad set of ligands and proteins interactions. So in that sense, if you have some what's called K-off data, so if you have some K-off data in the draw, preferably with structure data on your protein, then we would be very interested in running that and proving that what we have done actually works for what you're looking at. And from there, we can push it into existing projects where we would actually add value to the current drug discovery active projects, you can say.